Hi, I'm Niall Mangan. I'm an assistant professor in engineering sciences and applied math at Northwestern University, and I am a scientific leader at the National Institute for Theory in Math and Biology. My research started out in this area of thinking about how do we build models that will help us understand biological systems that have complexity and how they organize things in space and time. The type of models I'm interested in building are dynamical systems models. And that means that we understand how different populations or concentrations of chemicals or any other species of anything, whether that's chemical or like cells or whatever level, how they are interacting with each other in time. The traditional way we build dynamical system mathematical models to describe these things, it's also very slow. The methods that we've been working on more recently are how to automate that. One of the challenges for AI and ML is how to make it scientifically aware of the things we already know. And how to make it so that it is reliable in a way that doesn't necessarily matter if what you're trying to produce is a poem compared to trying to produce a scientific hypothesis. My group now thinks about what are the constraints of what kind of data you can get from biology, and what do those constraints on what kind of dynamics you can measure influence how well you can construct a model using an automated process. One of the things I've been really excited about being at NITMB is that I'm talking with people who have more background in optimization and things like algebraic geometry and algebraic statistics, which are, to me, really cool tools to try and understand how we should do computation better on the optimization side, but also how we can set up our problems so that we will comprehensively understand the models that come out. A lot of my collaborators are synthetic biologists, and what that means is that they learn about the natural world of biology and then they use that knowledge to build new things and solve societal problems. And so some of the projects that I've been involved with them on, in particular, has applications in green chemistry and being able to produce new products. I'm specifically interested in biofuels, but there's lots of things that we can potentially produce if you can better control the chemical reactions that are happening in cells. During COVID, I got pulled into a testing diagnostics project where the goal was to try and make a point of use COVID test. The collaborators I have gone on to rebuild that as a plant viral detection system. And they work with farmers, primarily in Kenya, to figure out what do we need to do in order to be able to deploy point of use virus detection method in places where farmers really need to know information about what viruses that they have out in their field and currently have no information until the plants are dying. Another application area is I have some friends, again, in synthetic biology who think about how to program mammalian cells to respond to signals from your body and then produce some signal or product or something that will help modulate what's happening. So a key example would be cells that you would inject into a tumor, and then they could tell how active the cancer was and put out stuff to shut that down. I have been really lucky to be involved in NITMB since we were writing the proposal for it. You know, the thing that was appealing to me was collaborating with biologists and being a part of an institute that was going to support those collaborations. Having a place that I can bring my existing biological collaborators and say, hey, here are other people who are excited to learn about and think about your problems is just a lot of fun.